So far you've had a look at a gene producing a protein, but in actual fact one gene is able to produce many different proteins. And we're going to have a look at the reason why that is. And the reason that one gene can produce many different types of proteins is due to this process here called alternative RNA splicing. Now you learnt before that when the primary mRNA transcript is produced, the introns are removed from that and the exons are spliced together to make the mature transcript. Now, under certain conditions, alternative segments of the messenger RNA may be treated as exons and introns. Well, what do I mean by that? Now let's have a look at this diagram on the next slide. Here you'll see that we've got our primary transcript. Now, depending on which exons are retained, you can produce different proteins. So you'll see from this primary transcript that is here, on this side over here, these exons here, here, and here have been retained, and that, when it is translated, goes on to produce this protein here. But in this situation over here, what has occurred is that these exons here have been retained, and they're different from the ones that were over on this side over here, and that results in a different protein being formed. It's important to notice that the actual order of the exons doesn't change in this process. It's just that different exons are retained. So different mRNA transcripts can be produced from the same primary transcript depending on which exons are kept. In other words, one gene can produce several different mature mRNA transcripts, and that means that different proteins can be expressed from the one gene. So let's go on and have a look at these proteins that are produced as the result of gene expression. Now, the proteins that are produced determines the phenotype of an individual, and environmental factors can also have an influence, but the major influence there is the gene expression. And the proteins that are produced will have a certain structure and function, and that structure and function will be dependent on the order of the amino acids that make up that protein. And the order of those amino acids will form the shape of the protein. So how does that occur? Well, here you'll see we've got our polypeptide here, and along that polypeptide there will be certain amino acids. There's 20 different amino acids that are involved in making up the different polypeptides, and we know the order of those amino acids is determined by the order of the bases on the DNA code. Now, you might have in this situation here a polypeptide that's got an amino acid 1, we'll call it, and 2, 5, and 6, 7, and 8. Now what happens is these amino acids have got different chemical structures, and that can result in amino acids. They're linked together by your polypeptide bonds, remember? But they'll have these different chemical structures, and certain chemical structures may be attracted to each other. So for example, this part here with this amino acid here may be attracted to this amino acid here, and that results in a folding situation. So here you'll see there's some folding that's happened here on the polypeptide chain because of something called a hydrogen bond. That is an interaction that's occurred between the amino acids on this part of the chain here and this part of the chain here, causing the polypeptide chain to start to fold. And this happens all along the polypeptide chain, and the polypeptide will form a three-dimensional shape, giving the protein its specific shape for that order of amino acids. In this situation here that I've highlighted, then it's held together by hydrogen bonds. There are other interactions that happen between individual amino acids, and these are named on here, but you don't have to know these other names. So the protein folds due to these various types of chemical bonds. You may have a situation where you've got a different polypeptide, or you will have a situation where you've got a different polypeptide, and there are different amino acids that are along here. So you'll get different interactions that are happening. So you may get the protein folding, or you will get the protein folding in a different way due to those interactions. And this gives the proteins that are produced a large variety of shapes, all of which have a different function. So this protein that's produced here will have a different function to the protein that's produced here, and that is all linked to the shape that the protein has.